A very good evening, everyone, and welcome to our first online series of lecture on the techniques in plant molecular biology. Today we will start with the basic techniques in gene cloning. So, we'll start with gene cloning. So, what is gene cloning? So, as we know, the cloning technique means to make multiple identical copies of the gene. So, gene cloning means we have a fragment of gene and we have to make identical copies of that gene. Now, the first terminology that comes under this chapter is the recombinant DNA. So, what is a recombinant DNA? A recombinant DNA is any artificially created DNA molecule that has been synthesized by bringing together two or more different DNA fragments from two or more different sources. So basically, we are synthesizing a DNA molecule by joining together fragments of DNA from two or more different sources which are not normally present together in nature. Now we must remember here that this phenomenon is nothing new. This recombinant DNA is always produced inside the cell during meiosis, and this recombination occurs during crossing over state. So we have just utilized this concept from nature, and we have utilized this concept to produce a chimeric recombinant DNA molecule according to our interests. So the next terminology that comes here is a recombinant DNA technology. So what is a recombinant DNA technology? In a very simpler way, this is a technology that helps us to synthesize recombinant DNA molecule. So what basically happens, we first have to select the gene of interest and we will join together this DNA fragments from two or more different sources, and we will then introduce this recombinant DNA molecule inside a host cell, and this host cell will now produce a new genetic combination, and this genetic combination will be of value to science, medicine, agriculture, and many other industrial purposes. So now we must understand that why is this gene technology important? So this recombinant DNA technology will enable us for three main purposes. What are this? First, we can produce a particular chemical product inside the organism. So we can make an organism to synthesize a particular chemical product in more amount. For example, we know that penicillin is obtained from Penicillium, a fungus. So we can manipulate this fungus to synthesize more penicillin for us, because penicillin is economically important. Second, we can prevent an organism to synthesize a particular chemical product. For example, lathyrus is a legume which is very nutritious otherwise, but it contains one neurotoxin which is called beta-ODAP. So because it contains this neurotoxin, it is not edible to us. So if we can stop synthesis of beta-ODAP in lathyrus, this legume seeds will be edible. So this underutilized legume can be utilized economically. And the third way we can use recombinant DNA technology is to enable an organism to produce an entirely different product. For example, insulin is one of the most sought after medicine that is used for treatment of diabetes. We all know that. So we can use some organisms, say for example yeast, to synthesize insulin for us. This is called bioreactor. So we can apply this recombinant DNA technology to synthesize this insulin inside yeast, and yeast normally do not synthesize insulin. So we can make this yeast to synthesize a completely new molecule for us. So having said that, we need several tools to clone a gene and to synthesize a recombinant DNA molecule. So what are these tools? Now the first thing that we need is some molecular scissors, so that we can cut the DNA fragments. And we need to cut this DNA fragments at specific sites where we need to cut them. And this molecular scissors are the restriction endonucleases. So this restriction endonucleases are the enzymes which serve as molecular scissors in gene cloning. Now, once we have cut the DNA fragments, we need to stitch them together. And for stitching them together, we will need some glue. So this glue in molecular biology is the DNA ligase enzyme. So we have the scissors, which are the restriction endonucleases, and we have the glue, which is the DNA ligase. Now, once we have synthesized this recombinant DNA, we need to introduce them inside the host cell. And for that, we need to have some vehicles for transportation. And these vehicles are the vectors. Now, these vectors include plasmids, bags, yolks, and so many other options. So, we will learn about this vector specifically in our subsequent classes. The fourth important tool is the gene library. The gene libraries are the repositories where we have all the genes of the organism present, from where we can select which gene of interest we have to take. And the fifth and final one is a strategy so that we can select which of the transformed host cells actually harbor our recombinant DNA. So we have to select that among say 10,000 host cells that has been transformed, which cells actually harbor our DNA. So for that, we use some selectable marker system which will help us to select the genes at the host cells that are harboring our RDNA. So with these 5 tools, we can now move on to clone a gene. Now let's zoom into all these tools one by one. So the first one is the restriction endonuclease. So what are restriction endonucleases? This restriction endonuclease or restriction enzymes are the molecular scissors in gene cloning. And these molecular scissors were first discovered in bacteria in 1962. Now, this discovery was so remarkable that the Nobel Prize in 1978 for medicine and physiology was shared by Werner Arber, Hamilton Smith, and Daniel Nathans for the discovery of restriction enzymes. So you can understand that how remarkable this discovery was. Now, these restriction enzymes are basically some bacterial enzymes that are involved in their immune system. Till date, more than 3,000 enzymes have been identified and they have several unique properties. So what are their unique properties? These enzymes can cut double-stranded DNA molecules in a sequence-specific manner. So they have a specific sequence where they will specifically cleave the DNA molecule. Now, because we have identified so many different restriction enzymes, we need to systematically name them. And because there are so many enzymes, we first need to lay down some rules so that there is no ambiguity in the nomenclature. For restriction enzymes, we have a very simple set of nomenclatural rules. What is that? Now let us see this example. Hint 2. Hint 2, this enzyme was isolated from Haemophilus influenzae strain D. So we will name it as the first letter H in capital will come from the first letter from the genus name. So H stands for Haemophilus. The second and third letters in small letter represents the first 2 letters of the specific epithet. That means in from influenzae. The fourth letter in capital represents the strain. In this case, it's the strain D. And then we have a Roman numbering which represents the order in which the enzyme has been isolated. That means hint 2 is the second enzyme to be isolated from Haemophilus influenzae strain D. Similarly, EcoR1 is the first enzyme to be isolated from Escherichia coli strain R. So in this manner, we can systematically and very easily apply names to all restriction enzymes isolated. So once we have named them, now let us see what are the specific features of the restriction enzymes. These restriction enzymes are bacterial enzymes that can specifically recognize sequences of 4 to 8 base pairs long. These sequences are called the restriction sites. And the restriction enzymes can cleave both strands of DNA specifically at this restriction site. So the most important advantage here is the cleavage is always at a specific sequence in a site-specific manner. And why do we call it restriction endonuclease? Because these enzymes will cleave DNA within the molecule. So we call it endonuclease. Now all these endonucleases have three main functions. These are recognition, cleavage, and modification. Now let's have a look at this picture. The enzyme first scans along the double-stranded DNA and looks for the restriction site. Once it encounters the restriction site, it will recognize the sequence and then it will cleave both strands of DNA within this recognition site. So the cleavage is exactly site specific. We can exactly predict where this enzyme will cleave the DNA fragment. Having said that, we must wonder that how does E. coli, why does E. coli have this enzyme in the first place? This bacteria basically use these enzymes for their immune system. These enzymes in E. coli will digest every foreign DNA particles that has been introduced into the cell. So once the bacteriophage has introduced their DNA molecule, they are foreign to the cell, and so this restriction endonucleases will cleave this top of this phage DNA. So the phage DNA cannot infect them. But have you ever wondered that how it protects its own genomic DNA? Because the genomic DNA might also have restriction sites, but if it chops it up, its own genome will be degraded. So that they must come up with some idea how to protect their own genomic DNA. For that, they have developed a restriction modification system. That means for every restriction enzyme they have a coupled methylase enzyme. So every restriction endonuclease has a corresponding methylase activity. What it does, this methylase enzymes will recognize the same restriction site as that of the endonuclease enzyme. And once it has recognized the site, it will specifically methylate one or two residues in this site in a site-specific manner. For example, in case of EcoR1, the methylase will methylate this two adenine residues specifically. And once methylated, the restriction endonuclease will be won't be able to digest this sequence. So E. coli uses this methylase enzymes to specifically methylate all the restriction sites in its own genome, and once methylated, the enzymes cannot cleave them. So in that way, they protect their own genome from cleavage. But the phage DNA is not methylated. So once the foreign DNA is introduced inside the bacterial cell, this enzyme will chop off the foreign DNA, and thus it will save the bacteria from phage virus attacks. Now, we must remember that for molecular biology, we do not need this methylase activity, because if the sequence is methylated, the endonuclease won't be able to chop them off. So, of course, we need to remove or eliminate this methylase function, and we need to select only the endonuclease part. Now, let's look at the endonuclease system. All the restriction endonucleases can be grouped into four main categories. These are the type 1, type 2, type 2S, and type 3 classes. Among them, the type 1 and type 3 enzymes have bifunctional enzyme. That means it has 2 subunits or 3 subunits, and they have both the functions together in the same enzyme. So these groups of enzymes cannot be selected for molecular biology purpose. Type 2 and type 2S enzymes have separate endonuclease and methylase activity. So for these enzymes, we can select the endonuclease part and leave out the methylase. But in case of type 2S enzyme, the cleavage occurs on one side of the recognition site, but away from the sequence, up to 20 base pair away from the sequence. So the exact site of cleavage cannot be predicted. But in case of type 2 enzyme, the cleavage occurs exactly inside the restriction site. So we can specifically predict where the cleavage will take place. So this is of real advantage, and so for molecular biology, we always select the type 2 restriction endonuclease. Now let us see what are the features of this type 2 restriction endonuclease. This type 2 restriction endonucleases are simple enzymes that have separate endonuclease and methylase activity. And of course, for our purpose, we will select the endonuclease and leave out the methylase part. These enzymes will recognize a hexanucleotide sequence. That means the restriction site will be 6 base pair long, and the cleavage will occur specifically within this restriction site. And these enzymes will require only magnesium ion as cofactor, so preparation of buffer is also simple. These enzymes can be of two types. The first category will chop off the sequence just at the middle of the restriction site, and they will cleave both the strands of DNA at the same point. So we have two blunt end fragments. The resultant fragments will have blunt end. Just like this, they will cleave over here. Both the strands will be cleaved over here. The second group will produce staggered end or sticky ends. In this case, cleavage will occur inside the 6 base pair region, but at different sites. So one strand will be cleaved over here, the other one is here. So for both the fragments, we will have a short single-stranded overhang, and the two fragments will have two overhangs that will be complementary to each other. So if you leave both the fragments together, these complementary overhangs will base pair and stick together, and that gives the name sticky end. So example of the sticky end enzyme is EcoR1, and one of the enzyme that produces blunt end is PVU2. Another example is MA1. So these are very commonly used enzymes in molecular biology labs. Now this is some of the examples: EcoR1, HIN3, BAMH1, HAE3, SAU3A. These are some of the commonly used restriction enzymes, and some of them produce blunt ends and others will produce sticky ends. For example, EcoR1, HIN3, BAMH1 and SAU3A will produce sticky ends, while HAE3 is another blunt end producing enzyme. So what are the applications? This restriction enzymes can be used for gene cloning, obviously, and apart from that, they can be used for RFLP analysis, DNA sequencing, synthesis of gene libraries, synthesis of gene chips, and so on. But in this lecture, we will concentrate mainly on the role of restriction enzymes in gene cloning. Having said that, let's look at the practical purpose. This is one typical digestion reaction condition and composition that we use while digesting DNA in the lab. What we need our DNA fragment that we need to cleave. Then we need water, of course, and please keep in mind that this water must be nucleus free, because if there is a DNA contamination here, this DNase will chop off our DNA template. So the water must be nucleus free. Then we will need the optimum buffer, and this reaction buffer must contain magnesium chloride. In some case, we need BSA. For some enzymes, BSA is optional and it depends on which enzyme we have selected. And of course, we need the restriction enzyme. Now, after adding all these components appropriately, we will incubate this mixture, this reaction mixture, for about 1 hour, 1 to 1.5 hours at appropriate temperature. The most commonly used temperature is 37 degrees. So after incubation, we will heat inactivate the enzyme by incubating the reaction mixture at 65 degrees centigrade for 20 minutes so that the enzyme gets inactivated. So this is how we typically digest a DNA in the lab. Next is double digestion. So what is directional cloning and double digestion? We all know that in order to express a gene, we need to place them inside the
expression cassette. So what is an expression cassette? Expression cassette means we need to put the gene in such a way so that it has a promoter in front of it and a terminator at the end of it. So we have to make sure that the gene is placed in the correct orientation and it doesn't get reversed. If it gets reversed, the sequence will be read in the opposite direction, and that will lead to the synthesis of wrong protein product or truncated protein product. So we must ensure that the gene of interest is inserted in the proper order, proper direction. So for that, one of the easiest option is to use two different restriction endonucleases at two different ends of the gene. So we have to select two restriction enzymes such that it is present in the vector but not inside the gene. If the restriction endonuclease site is present inside the gene, it will chop off the gene from within, which is undesirable. That will destroy the gene. So this selected restriction site must not be present within the gene. And let's see here, we have EcoR1 site which is present just after the promoter. So we can place this EcoR1 at the beginning of the gene, and we have NOT1 site just before the terminator. So we can place this NOT1 at the end of the gene. Now we will digest both the plasmid and the gene of interest with this RE pair. So it will produce a staggered end or sticky end at two ends of the gene of interest and also at two ends of the digested vector. But because we have used two different restriction enzymes, the single-stranded hang over here is not similar to that of here. So the digested plasmid or digested vector will not self-ligate. These two ends cannot base pair. Similarly, this end, the EcoR1 end, will produce a overhang that can base pair only with this EcoR1 overhang. And the NOT1 overhang will base pair only with the NOT1 overhang in the plasmid. So the base pair will always be directional, and so we will always have a directional clone. So we will always select two different RE pairs while cloning. Now please remember that if we select blunt end restriction sites at both the ends, there is always a chance to get self-ligated products, and also the directional nature will not be maintained. So the easiest way is to use two sticky end enzymes or one sticky end and one blunt end enzyme. This is the easiest way. But we can always use two blunt end enzymes, but in that case we have to phosphorylate and dephosphorylate the ends during ligation to ensure directional cloning. So that will add up some extra steps in our cloning. So for most practical purpose, we either use two sticky end enzymes or one sticky end and one blunt end enzymes. Next, having understood all details of this restriction enzymes, we will come across these two terminologies: isosisomers and neosisomers. So what are isosisomers? Isosisomers are just like isomers. These are basically restriction enzymes that have the same cleavage site and same recognition site. So for example, SPH1 and BBU1 has the same restriction site. That means they have the same recognition sequence and same cleavage pattern. So they are called isosisomers. Another similar case is that two enzymes might have the same recognition sequence but different cleavage patterns. These groups are called neosisomers. See, for example, XMA1 is an enzyme that has the same restriction site, same recognition site as that of SMA1. But here, the cleavage pattern is staggered, and in case of SMA1, it is a blunt end cleavage pattern. So the cleavage pattern is different, but recognition sequence is same. These pairs are called neosisomers. Okay. Okay. The next thing is that star activity. Now, in all molecular biology labs, this is one of the most problematic phenomenon that we face during digestion reactions. So what is star activity? Star activity is basically the non-site specific cleavage by the restriction enzymes. So this is a phenomenon that leads to non-specific digestion by restriction enzymes. Now, why do these enzymes exhibit star activity? One option is that we have used too high concentration of glycerol, so the optimum reaction condition is not maintained. The second thing can be high concentration of enzyme has been used, so the enzyme is to substrate ratio is not mentioned. The correct buffer has not been used. That can also happen. Or we have incubated the enzyme and the DNA together for a prolonged time. So in these cases, the enzyme will show non-specific cleavage activity, and we will end up with a smear with a totally chopped up piece of DNA, which is undesirable. So, of course, we can work to minimize these problems. We can use lower glycerol concentration. We can use lower enzyme concentration. Exact buffer conditions should be maintained. And the incubation time must be restricted to 1 to 2 hours and not more than that. Having said that, there are some restriction enzymes which has a higher tendency to show star activity than others. So, by selecting our restriction endonucleases in cloning, we must take care that the enzyme must have a lower tendency towards star activity. And these informations can be easily available from the manufacturer's website. All these commonly used restriction enzymes are commercially available, and they are supplied along with their buffers and the reaction conditions. So we must take care that we are using the exact buffer and optimal restriction digestion in incubation conditions. So in that way, we can prevent star activity. Okay, so the next tool, we must now focus on the next tool in molecular biology, and that is the DNA ligase. A DNA ligase is the glue in molecular biology. It is an enzyme that can catalyze formation of a phosphodiester bond between the 3' OH of one fragment and the 5' phosphate of the other fragment. So it will stitch the fragments together. Now we have encountered this DNA ligase during replication procedure where the Okazaki fragments are stitched together by DNA ligase. The same enzyme is used here to seal the fragments together. And this reaction in which the DNA ligase catalyzes formation of a phosphodiester bond is called ligation reaction. Okay. So we have two main groups of DNA ligases. Here we can see that DNA ligase catalyzes the reaction in which a phosphodiester bond is formed between the 3' OH of one fragment and the 5' phosphate group of the other fragment. And this results in joining together of the two fragments. Now we have two groups of DNA ligases. One group is isolated from bacteriophages or mammals, and this bacteriophage DNA ligases requires ATP as cofactor, and they can ligate both sticky ends as well as blunt ends. The other group are the bacterial DNA ligases. They use NAD+ as cofactor and can ligate only blunt ends. So of course you all can understand that for practical purpose it is easier to use bacteriophage DNA ligases because the same enzyme will be able to ligate both sticky ends as well as the blunt ends. So in molecular biology we all use bacteriophage ligases, and the commonly used one is the T4 DNA ligase. So this T4 DNA ligase catalyzes the synthesis of phosphodiester bond formation between the 3' hydroxyl group of one fragment and the 5' phosphate of the other fragment, and it requires ATP as its cofactor. Now once the ligation reaction is over, you can inactivate this enzyme by heating it to 70 degrees centigrade for 10 minutes. That's how we inactivate the enzyme. All right. Now let's have a look at a very commonly used condition, how we set a ligation reaction. A ligation reaction requires several components. The first one is, of course, our digested vector and the digested insert DNA. And we must keep in mind that both the vector and the insert has to be digested with the same pair of restriction enzymes. If we are using sticky end enzymes, if we are using blunt end enzymes, different enzymes can be used. So we have vector DNA and insert DNA that has already been digested. Then we have to use the proper buffer. This is called the T4 DNA ligase buffer. We use nucleus-free water to make up the volume, and we use the enzyme T4 DNA ligase. After we have set up this reaction mixture, we will incubate the mixture either for 16 to 18 hours, that means overnight at 16 degrees centigrade or 4 degrees centigrade, or we can incubate the mixture for 2 hours at room temperature. Okay, so this ligation reaction is one of the most tricky step in gene cloning because this is a chance factor. So for every cloning step, we need to optimize the proper ligation reaction condition. And while optimizing this ligation reaction, we must keep in mind that we can look at 5 different steps. What are they? First and most important one is the molar ratio between the vector and the insert. There is a wide range of ratio that we can use, and that may vary from 8:1 to 1:16, which is huge. But the most commonly used ratio is 3:1 or 1:3 or 1:6. But again, we have to optimize this ratio according to our case, our insert and our vector. Next, we must use 10 to 200 nanogram of plasmid. If we use more than that, ligation efficiency is minimized. The volume of reaction is usually 10 microliter and we do not alter it. And the fourth one is the amount of ligase. We start with 1 unit and then we can take this amount increased up to 10 units according to the quality of the ligase, of course. And the fifth one is the incubation time. Now, we must keep in mind that if we are using blunt end ligation, it can be performed at 4 degrees centigrade overnight. On the other hand, for sticky end ligation, we can incubate it at 4 degrees for overnight, or 16 degrees for overnight, or 2 hours at room temperature. Again, the most commonly used temperature is 16 degree or 4 degree and overnight. That has the highest efficiency for ligation. So with that, we will end our class today. In the next class, we will learn about the DNA modifying enzymes. Apart from the ligase and the restriction endonucleases, there are several other DNA modifying enzymes. We'll learn about that, and we will learn about the different types of cloning vectors that we can use. So till then,